So our next speaker is Dr. Channing Sebo, and what she's going to be talking about is vaccine technologies and tracking emerging diseases. And we thought this topic might be pretty interesting to everybody because of the COVID pandemic that we're having. But there's some pretty interesting technology out there working around vaccines and vaccine technologies. But more than that, there is some interesting technology about tracking and creating heat maps for emerging diseases. So that's the reason we kind of wanted to have Dr. Channing on. But anyway, she is a 2015 graduate of Iowa State University. That's where she got her DVM degree. She has the managing veterinarian or veterinarian and diagnostic analyst with Sebo-Decker technology, which makes different swine vaccines for us. In her role at Merck Animal Health, she works with Sebo-Decker technology since 2013, leading the field and expertise in vaccine design and implementation in swine. She created and developed Sebo-Decker dashboard, bringing disease tracking and vaccine design to veterinarians in the field. So with that, will you start her video, Dr. Epperson?
Today we're going to talk about vaccine technologies and tracking emerging diseases. My name is Channing Sebo-Decker, and I am a diagnostic analyst and swine technical service veterinarian for the Sebo-Decker technology with Merck Animal Health. I grew up in Wisconsin, and even though I am a swine vet now, I grew up in dairy country and on hobby farm, so with tons of animals, equine and dogs and cats and whatever other animals I could bring home. Definitely grew up in Wisconsin, so a huge Packer fan. And I went to undergrad at the University of Minnesota, graduating with a double major in animal science and biology. In 2011, I received my DVM from Iowa State in 2015. While I was in vet school, I started working with Harris Vaccines, which is the former company of the Sebo-Decker technology, and as an intern. And throughout vet school, I stayed with the company. When I was graduating in 2015, I started on as their tech service veterinarian. Merck purchased the Sebo-Decker technology from Harris Vaccines in 2015, in the end of 2015, and I stayed on with the technology since then. We developed the Sebo-Decker dashboard as a way to track and analyze the sequences and also track and manage evolving strains as well as pathogens. And I've been a part of that basically ever since. So today, we're going to use heavily the RP or Sebo-Decker technology to learn about different vaccine technologies and how they've evolved over time, as well as I know everyone's wondering how the COVID-19 vaccines relate to our current technologies as well as the RNA particle technology. And then I also want to highlight different methods that we have for tracking diseases as well as sequence analysis, especially tracking emerging strains and different evolving strains over time. So looking at the evolution of vaccine technologies over time, starting with our first generation vaccines. These encompass the majority of the vaccine products we have available for our use today in animal health. These are our traditional vaccines. Starting with first, the inactivated or killed vaccines. Autogenous would also fit into this category. These biologics have been rendered non-pathogenic or not capable of causing disease. Most have been combined with an adjuvant to stimulate a necessary immune response. We can sometimes see vaccine reactions associated with these adjuvants. The other major group in our first generation is our attenuated pathogens or our modified live vaccines. These pathogens have been treated, have gone through passages, or modified in some way to reduce virulence of the pathogen. They are still alive. Most will still replicate, and at which level and how much they can replicate and how much they have the ability to cause disease is variable based on the method, the pathogen, and also the immune status of the animal being vaccinated? Are they immunocompromised, or are they challenged in some other way? These are all things to consider when designing the protocols as well as selecting vaccines. It's important that because they are alive, we do get a robust immune response from these vaccines, and they will generate both a cell-mediated as well as a humoral immune response. From a research and development side to licensing timeframe, these first-generation vaccines take years to develop, and overall, these first-generation vaccines are well established and trusted in our industry as well as in human health. Second-generation vaccines make up our purified antigen and subunit vaccines. This is in contrast to the first generation vaccines, which are whole organism vaccines, as we just discussed. Recombinant proteins, conjugates, or inactivated recombinants. These might use expression vectors such as E. coli, yeast, baculoviruses, etc. So last, but certainly not least, is now our third generation vaccine technologies. So this would be our vectored genetic material vaccines. So this would be DNA vaccines, pox, live chimeric, as well as what we're going to learn most today is the non-replicating RNA particle, or RP, vaccines, which is the Sequivity technology. And I'm going to use this technology that Merck has to showcase how these third generation vaccines kind of combine technologies and advantages of the traditional vaccines that you are all familiar with and use every day, and how those kind of can encompass and leverage different components of the immune system as well as manufacturing efficiencies. And then we can apply that in how we're fighting COVID-19 and the mRNA vaccines that they're having. One important note when we're looking at these vector genetic material vaccines is that one of the challenges that why this technology has not been widely absorbed is that a lot of the issues that have come into play is that the animal itself will develop an immune response to the platform, the vectored vaccine technology itself. And that is an issue with DNA vaccines, pox vaccines, as well as live chimeric vaccines, is that the animal, after repeat injections, develops an antibody response to the vaccine itself, not necessarily the antigen you're trying to get it to respond to. And that is something that sets the RP vaccines apart from the rest of the technologies in this group. The mRNA vaccines, I included it in the third generation because that's a new technology. I don't know yet if it's going to be a third generation or if it'll actually be a fourth generation of vaccine technologies. So it's kind of an unknown. It hasn't yet been grouped in. So I have it kind of as a separate side note right now. But I'm going to kind of show you how it pairs into it. It actually has quite a bit of a different technology that we're going to use, but I'll show those as we go through. So looking closer at the RNA particle vaccine technology, this technology is based on an alphavirus. And the alphavirus that it's based on is Venezuelan equine encephalitis virus, or V. Alphaviruses are single-strand enveloped RNA viruses, and it is a platform vaccine technology. And what I mean by that is that this platform has been created, and regardless of whatever pathogen we are going to make vaccine for, our manufacturing process stays the same, and we use the same platform regardless of the pathogen we're targeting. So the way the platform's been modified is we've taken this V virus and we removed these structural genes. And the structural genes that were removed are the capsid and the envelope genes, the E1 and the E2. And that's really important because that's what makes the platform safe, by making the platform replicative defective. So it has no ability to replicate in the animal, which means it has no ability to cause disease or recombine into a new virus. This alpha virus is simply our carrier virus. There is no risk for infection or no risk for the ability for it to cause disease. This same platform, again, is used for all of our vaccines. So getting into the manufacturing. So here's our platform again, a schematic of it. So here we have our non-structural genes that remain in the platform, and we're going to insert a gene of interest. This gene of interest is a genetic sequence, and this is going to encompass just our A, T, C, and G, the genetic code of the pathogen we're targeting. And in the next slide, I'll get into what a gene of interest is. So we're going to take this platform, which is also called our replicon RNA, and we're going to combine it with helper RNAs, which is our glycoproteins and our capsids. We're going to combine those with vero cells, which are our cell lines, and we're going to electroporate those. And electroporation is just a fancy word for a shock. And what that shock and that electric charge does is it opens up the pores on the vero cells, which are our cell line, and it allows a copy of each component that we need in our manufacturing process into the vero cells. We then let those cells rest and then put those into roller bottles for incubation. Those are then incubated for 18 to 24 hours, during which time the vero cells make tons and tons of copies of our RNA particles. And those are the gold little figures you see on the bottom. And if we zoom in on those gold figures, on the outside, the gold balls look like alphaviruses or V particles, and they're going to look like an alphavirus or a V to the immune system. So that's why we get the immune presentation of a alphavirus. But on the inside, there is just the non-structural V genes along with our gene of interest. This makes the RPs themselves replicative defective and without the ability to cause disease. As we saw in the previous slide, the gene of interest is a gene or a genetic sequence that we're going to insert into our platform. A gene of interest is a sequence for a pathogen that is a gene that we found to be antigenically stimulating to provide protection against that disease. Examples of genes of interest, or GOI, are glycoproteins, toxins, receptor molecules, or structural proteins. So some examples that we're currently using in our platform would be for influenza, would be our most common. This would be primarily in swine, but it could have applications in canine, equine, as well as other species. This would be the HA or hemagglutinin protein. We also heavily use the rotavirus, A, B, and C, the VP7 gene. And one I'm going to focus on heavily today would be our PED, or porcine epidemic diarrhea virus. It's a coronavirus, and that would be the spike gene. And then we have a few others here that are listed. So I'm very much a visual learner. So I thought I would include some picture representations of our manufacturing process to try to take a very microscopic concept of how vaccine is actually made from this method, and try to make it make a little bit more sense. So it's done on a very small scale, and I think that's very in contrast to any other manufacturing that's done for a lot of traditional vaccines. So in this picture, you guys can see that this is how small scale we're using to make vaccines. So in this picture, these are small little cuvettes. They look like tiny little test tubes. This vial, it will make 10 to 20,000 doses of vaccine. So this is actually mixing the replicon, which contains our gene of interest, along with the V non-structural genes. And we're mixing in our vero cells along with our helper RNAs into this. This is our electroporator. So that's the electrical charge that's going to be used to open up the pores of the vero cells. So we're going to take those cuvettes and we're going to put those into the electroporator. You can see on the side of the cuvettes, there are metal plates. And their metal plates are located on both sides of the cuvettes, and that allows the electrical charge to pass through the cuvettes, which allows that electrical charge to transfer through the virial cells, opening those pores, allowing the copies of the platform along with the helper RNAs into the cells. We're then going to rest those cells for a few seconds, and then we are going to seed those cells into our roller bottles into a media. And then these roller bottles are then placed in incubators. And these are roller bottle incubators. And these incubators just take those roller bottles and they roll them continuously for 18 to 24 hours. What that does is the media, along with the virial cells that have the copies of all of our manufacturing components, seeds the outside of the roller bottle tubes, and it gives a large surface area of the cells to grow on. And that then creates a surface area for the virial cells to grow. And when the virial cells are growing, they're going to then make a bunch of copies of all of our RNA particles. And that is our component that we then can harvest, and that is our vaccine. So when we go to harvest, there are two different harvest methods that we're going to use. One is a filtration. So we know the size of the RPs, so we can use different filtration methods to capture the RPs themselves. We can also use different salinity changes due to the charges of the RPs to be able to capture the RPs in a certain solution. And this process is to capture one single strain of ARP. When we're going to then make different combinations of vaccines, so we can create multivalent vaccines, so different strains, so we can combine different pathogens all into one bottle. We can then create those multivalent vaccines by repeating this process for different strains. And then we get to formulation. So we then take all of those different RPs and create those into our formulation. So we're going to mix those basically, all the different RPs into the bottle and mix it with basically a saline, and then put those into our vaccine bottles. And then we ship those to our customer. Our entire manufacturing process for vaccines that have been made previously is about 8 weeks. And if we are starting from scratch, where we have never made that pathogen before, or it's a brand new strain, it is about 12 weeks. So in summary, we started with our platform. We inserted our gene of interest. So for example, for influenza, it'd be the HA, or for PED, it would be the spike gene. We electroporated those into our virial cells. We harvested our RPs, and we combined those into vaccine. So enough about all the manufacturing. Now I want to get into the immune response. How do we actually get the efficacy when we inject these RNA particles into the animal? So an important feature is that we can generate a robust and balanced immune response without the use of adjuvant. So we generate all three arms of the immune system: the innate, the humoral, which is your antibodies, as well as a cellular or a cell-mediated immune response. And we do that through the parent, the nature of the parent virus, the alphavirus. So alphaviruses, the V virus, has dendritic cell tropism to dendritic cells, just based on the nature of how they naturally infect. So when RPs are injected into the animal, they are taken up by dendritic cells and presented to the immune system. So we are basically using a natural behavior of the parent virus to trick the immune system into thinking that it's a live virus infection, but it's actually not, because we've removed those structural genes. It is just an RP that looks like a live virus on the outside, but it only contains the non-structural genes and our gene of interest. So by kind of secretly including our gene of interest on the inside, we sneak in an immune response for our gene of interest on the inside. We get high levels of protein expression for our actual gene of interest. 10 to 20% is a high level of protein expression to our target genes, our gene of interest. So we're going to get a lot of antibody produced directly to our gene of interest. So for influenza, that would be our HA gene. For our coronaviruses, that would be our spike gene. An important feature, when we had talked about those third generation vaccines, a lot of those, when you have a vector type vaccine, you see issues with when you give that same vaccine repeated number of times, the host actually develops an immune response to the vector itself. So it would develop a immune response to the V or the alphavirus itself. That is something that this technology does not have issues with. So we do not have issues with the anti-vector immunity. So that is a very cool and very nice thing. So we can repeat injections with the same gene of interest or different ones without having reduced efficacy. So some advantages. I think when we get into the coronavirus vaccine response, this is where a lot of this is going to become very evident. So when you are only requiring a genetic sequence for our gene of interest in order to design these vaccines, you don't require isolation, you don't require passages to make the vaccine into a modified live or a killed vaccine. The safety side of it is a lot less complicated as well. You can go from sequence in our technology from sequence to vaccine in a matter of weeks, or in these descriptions of what's going on with the COVID-19 fight, from vaccine to or from pathogen to vaccine in a matter of months. It has a very high safety profile, so it's considered non-replicating and non-living. When it's injected into the animal, it has a single round of protein expression, so it can't replicate, it can't cause disease, and most importantly, it can't recombine into new viruses, so it doesn't going to cause any risk of spread or have any issues with movement and recombine. It is DIVA compatible because we're only injecting the gene of interest. The rest of the genes of the virus would be available to test for. So an example of this would be for influenza. The HA gene would be included in the virus. An NP ELISA, which is a commercially available test, would be available to test for. If an animal would be NP ELISA positive, that would be due to a natural infection or a different type of vaccination. Another important feature from the safety component of it is no live pathogens ever enter our facility. Because we only need a sequence that is electronically sent to us via an email. So no live pathogens ever enter. So there's no risk of contamination of having to enter a pathogen into our facility. There's also no adjuvant in the vaccine. So we rarely to never see vaccine reactions. So no off-feed events, no abortions, things like that. So currently how the majority of our vaccines are licensed is through a veterinary prescription license. And this is in contrast to a autogenous license. So autogenous license are are very much licensed based on a killed vaccine, where you have to kill the pathogen, and it's typically mixed with an adjuvant. This prescription license is really a license that we worked very closely with the USDA CVB to create, and it's basically licensing a platform-based technology. So it's a customized prescription. It has to be a non-replicating, non-viable biologic product. We are only the current product available that is licensed under this platform. This license is granted based on a fully licensed product. So our fully licensed product is an H3N2 influenza product. It is a product that this license was generated based on one pathogen for one species. That licenses then can be granted across species for other pathogens. It basically outlines our manufacturing production. We can then apply as long as we have justification for our gene of interest, that we have an idea that there would be efficacy based on research of our own as well as from research labs, universities, etc., that gene of interest would be a good target for a new disease. It allows us to combat new and emerging diseases. It allows us to have this kind of plug and play concept. Currently, the majority of our research is in swine, and the majority of our products is sold in swine. For a little bit of history, this platform was originally developed by Harris Vaccines. In 2015, it was acquired by Merck Animal Health. And now research has gone rapid into all other species. And, slowly and surely through the next few years, we should start to have other products in other species. We also were limited to just U.S. sales and U.S. products. We've now gotten approval for different products in Canada and in Chile, and limited sales in other countries with our PED product. But those will slowly expand too. So getting back to how this platform compares to conventional, and then we'll get a little bit more into some other vaccine technologies. So if we think about back to kind of the basic components of vaccine technologies, we think about a master seed. So when we think about conventional vaccines, we start with our master seed is a virus or bacteria. When we think about it compared to these third generation technologies, and these are primarily sequences. And these are genetic sequences that we can get electronically that we're going to insert into our platforms. When we look at conventional versus this platform, for our antigen, this can highly vary in conventional. For the RP, it is a RNA particles with our gene of interest. For manufacturing, it requires growth of the pathogen with chemical activation typically, or passages. For the RP platform, it is identical every single time. We have the same identical workflow regardless of the pathogen we're targeting. From a safety standpoint, conventional is highly variable depending on the adjuvant, if it's killed, and if it is a modified live, then it is highly variable based on how that modified live was created, and the risk of virulence and or ability to cause disease. We have basically, limited to no reactions observed, and we have no risk of shed with the RNA particle platform. Development schedule of conventional vaccines, those first generation vaccines, those conventional vaccines take years and years. Typically they say 5 to 10 years at least for conventional vaccines to be produced. These third generation type vaccines, we're talking months to a few years. And typically a lot of that timeline is really in the regulatory standpoint, not really in the technology limitations. So these third generation technologies, I kind of like to think of them as we kind of get the advantages of the immune presentation of live products, where we generate the immune presentation of cell-mediated immune response along with our humoral antibody response, with the safety of killed products without the ability to cause disease and shedding a virus. And where we meet is kind of in these third generation, and the RP vaccine is the current example here. So one of the big issues we have in animal as well as human health is speed of production when we have new and emerging diseases. And when we look at traditional vaccine technologies where you have to be able to isolate a virus or a bacteria, typically viruses is what we're looking at for new and emerging. And you have to grow it up and modify it in some way and then find an adjuvant and do all the safety testing. We're talking years, many years before you get to the point of having a vaccine that is commercially available. And you have to do a lot with manufacturing techniques and whatnot. So when we look at these groups of third generation vaccines, you create a lot of efficiencies when you can start with a genetic sequence and go to a manufacturing platform-based technology that the vaccine is made the same way every single time, regardless of the pathogen that we're targeting. And so when you can create all of those efficiencies, you create your timeline basically cut down in greatly reduced. So I wanted to give you guys a few examples at how we've used this RNA particle technology to already tackle emerging diseases. And we all know that in the animal health industry, but also with COVID-19, emerging diseases are something that is hot on our radar at all times. So here is a great example and very topical is the porcine epidemic diarrhea virus, or PED. Back in 2013, we in the swine industry were hit pretty severely with PED. It's a coronavirus. Ironically, it actually came over from China as well, is where it originated from. It is a coronavirus. So coronaviruses, as many of you know, can primarily infect either respiratory or enteric. This is a coronavirus. In our side, this PED is an enteric virus. So it caused severe diarrhea with almost 100% mortality in young piglets. Less than 3 weeks of age, it was almost nearly 100%. It is highly infectious. It wiped out a lot of piglets for a very long time. And we immediately, when we were still Harris Vaccines at that time point, looked at vaccine options. And so we looked at what would our gene of interest be if we were going to tackle this pathogen with our technology. And here's the timeline of our vaccine formulation. So PED entered the U.S. in April of 2013. We immediately had a candidate vaccine for evaluation in May of 13. We had it out in the field by August of 13. So we had started laboratory trials by May, and we had it available in the field through our veterinary prescription in August of 13. So in a matter of a few months, we had vaccine in the hands of our customer to be able to try in their herds. Because of the safety of the vaccine and because of the basically desperation of our customers, when you're losing 100% of your pigs or 99% of your pigs, it's a pretty easy decision to try something. And many of our customers started to use the vaccine. It was given to sows pre-farrow after they had broken with the virus in most cases. By January, we actually had updated the vaccine. I'll go into it in a little bit when we talk a little bit more about the coronavirus, but the spike protein is actually split into two portions, the S1 and the S2 gene. Our first generation just contained the S1 gene. We found out that we actually needed the S2 gene as well to provide protection, so our second generation included the S2 as well. In January, we came out with our second generation, and within June, so a matter of just about 15 months after the outbreak occurred, we had a conditionally licensed vaccine out with mass production available to the entire U.S. population. And that is basically through an emergency condition, conditional licensees are granted. So this is a great example of how these third generation vaccines are available and can vastly reduce production timelines and research timelines to be able to combat, new and emerging diseases. A few other examples of where this technology was used to combat emerging diseases is with our avian influenza outbreaks in 2015. They were outbreaks primarily in Iowa as well as other states, and it was high path avian influenza. We received a sequence. Again, we didn't need to wait for any isolation of the virus or anything. We immediately made vaccine. We worked with the USDA as well as research partners to start efficacy trials. It is high path, so we can't just put vaccine out into the field like we were able to do with our PED. And so we were able to get a USDA stockpile emergency grant to be able to stockpile that vaccine for emergency use. And within a matter of months, we had millions of doses produced for that stockpile. We also did this exact same scenario with our PCV3 in swine in 2018, and that vaccine is widely used now today. Approximately 20 to 25% of the population in the U.S. is vaccinated with PCV3 in swine, and that was a disease that no one knew anything about a few years ago. So getting into COVID-19. So again, it's a coronavirus, an enveloped positive sense RNA virus. So here we're going to look at the spike protein, just like we did for PED when we were looking at swine PED. The spike protein is important because it has a role in the entry of the virus into host cells. The S1 portion of the spike gene is responsible for receptor recognition and binding, and the S2 is responsible for cell membrane fusion. So the vaccine technology that is currently the two that are furthest along in approvals are both mRNA vaccine technology or messenger RNA. This technology, the way I think about it and how it compares to like the RNA particle vaccine technology, is that it contains messenger RNA inside of a lipid nanoparticle. So instead of a piece of RNA enclosed into a RNA particle capsule that looks like a virus, it's enclosed into this lipid nanoparticle. Two companies that are working on this are Pfizer and Moderna. The differences between the two technologies are primarily in the technology of the lipid nanoparticle. That is what's leading to the different storage requirements. The target gene is, again, the spike protein. This was similar to the swine PEDV. What's great about this mRNA technology for the vaccine is, again, we can go without isolation of the virus to right to vaccine. So this creates a lot of efficiencies in R&D and vaccine development. It's allowed us to go from a vaccine to, basically trial in a matter of months, versus having to, grow up the virus, do different passages, different killing techniques, and all of those different avenues. It also has a lot more safety components of it. We're going to be generating both a cell-mediated and humoral immune response. This technology does generate both. And what I did a little bit of research into is also the mutation rates of coronaviruses. It is important to note that coronaviruses can go under some mutation rates. There has been some different mutation rates that have been reported. One article that I found, and I have some sources on the bottom here, is that the mutation rate of the current COVID-19 is about half of influenza. It's important to note, though, is that these vaccine technologies are only using the spike protein of the virus. So the mutations that are undergoing we only care about the mutations that are happening in the spike protein, not the rest of the virus. So those are the mutations that we're going to need to focus in on. And we only probably really care about the mutations that are causing actual amino acid changes. So the nucleotide changes don't necessarily matter unless they're causing amino acid changes, particularly if they're causing protein changes and different protein conformational changes. If those changes occur, then the proteins themselves are going to look differently and potentially have the antibodies to look different and potentially have lack of cross protection. Our last and hopefully a little bit lighter topic, we'll look at options for tracking emerging diseases and sequence analysis. So Merck's option is the Sebo-Decker dashboard. We developed and launched this dashboard in 2018 to analyze and track pathogens. Our first tool set that's available is the homology comparisons, and this is pretty much just a percent similarity comparison. So on the right, you'll see a homology chart, and this is just looking at a sequence to sequence level of how similar are they to each other. You can look at sequences, how they are to each other, as well as vaccines to the sequences or vaccines to vaccines and how similar they are. Our second analysis tool is a key site analysis. Key sites are specific amino acid positions within our gene of interest that have been shown to be very important for cross protection, or probably even more important for lack of cross protection. These key sites are typically in or around receptor binding sites, which are the regions that the virus is actually going to attach to the host cells and initiate this infection process. Where the virus attaches is going to be a huge target for vaccines, as well as the animal's natural immune response to make antibodies to protect and provide protection against that disease. Small changes in these key sites, say just an amino acid change from a positively charged amino acid to a negatively charged, or a hydrophobic to a hydrophilic, can cause major conformational changes in the protein structure in or around these binding sites. So if the area looks different, antibodies can't bind and recognize it. So they can't do their job to actually block infection. So in summary, these key sites, even with just a single amino acid change, so the sequence could be 99%, 99.9% on a homology comparison. Even these single amino acid changes, even within the whole genome of the virus, could cause critical missteps basically for the virus, and they're really, really important when analyzing sequences. We do have key sites pretty much understood for a lot of our influenza strains, and we're just starting work with rotavirus. The human side would also be doing a lot of work on the influenza, and that's where a lot of this research has come from. You can see on the figure on the right, we have our sequences listed to the left, and then positions 145, 155, etc., and then letters underneath. Those are actually the amino acids at those positions. So the dashboard actually extracts the amino acids at those positions. Those are our key sites. And then we can analyze the exact amino acid at that position, see which changes are occurring, and analyze it appropriately. Our third feature is a dendrogram, or a phylogeny. And I like to think about these basically similarly to like a family tree. So the closer they are, the more related they are. So we're going to use the distance of the horizontal bars on this graph to measure how related the viruses are to each other. The closer they are, the more related they are. So this is going to allow us to track how the evolutionary distance and how the viruses have changed over time to see where they're moving to and how they've evolved. Our fourth and final tool set that we have built into here is our geospatial or mapping tool. So this tool is probably one of the most used features. It allows us to map each of the positive cases along with sequence information and genetic information. So it allows us to watch each strain of positive cases, and it allows us to see where that case was positive with the genetic information behind it, follow it over time, as well as it spreads through an area. So we can find out the genetic information, find out where that strain is moving to, how many cases there are for it, as well as how much of an impact it's having. So this type of information and this type of dashboard allows customers to design high quality, relevant, custom vaccines for the animal health industry. Its current use is primarily in swine, but it has a lot of cross-species adaptations, especially for pathogens like influenza that are constantly evolving and changing. They're very diverse, and we're constantly having to update vaccines. We all know the human vaccine for influenza is updated every year. We do a very similar approach in our influenza vaccine. We actually update our vaccine 3 times a year. This also allows us to make regional specific vaccines. It allows us to track emerging strains and pathogens. It allows our customers to see, what's in their area. It allows them to make evidence-based scientific decisions. It allows them to look at, what's in their area, what's coming. The platform itself works with any and all sequences. So if a brand new disease that we've never heard of comes in, we can add it into the platform in a matter of, minutes to hours. It doesn't take new development. So it works with any and all sequences. I've actually started to play with a little bit of cattle stuff. I've looked at some BVD sequences, looking at the genetic sequences of those, the diversity, how much diversity is out there. We've started to look at some different key sites and some different diversity there. Not sure what it all means, but when we look at, different cross protections and where we have vaccine gaps and stuff, this is the kind of tool we can use to start to understand that. Our database grows more and more every day. We currently have over 39,000 sequences. So this geospatial analysis or the mapping tool is probably one of the biggest components that we use. It allows customers to answer the questions of what's coming into my area, where did this virus come from? It allows them to move from more of a reactive treatment type approach to a more of a proactive approach. And all of this data has remained confidential, so they can see what's in their area without knowing, what other customers are in their area. So it's all blinded. So here's an example of how we actually use this mapping tool to design vaccines and really track viruses. So this is an example for influenza. So in pigs, we heavily monitor the human side of influenza as well. So influenza can go from humans into pigs and from pigs into humans. So this strain we're going to look at here is actually a strain that has been commonly moving from humans into pigs. So on the top, there is a bar that is going to go over time. It starts back in September of 2018, and it's going to go through the end of August 2020, so just a few months ago. And when I press play, you're going to see little dots appear over the map, and each one of those dots is a positive case of this strain. And this is going to allow us to track the cases of influenza over time. We see that there's a lot of positive cases, and then in about April of 2020, we see an explosion. So this helps us show an indication of what this strain is doing and how it is moving throughout time. We also use the other tools in this dashboard to analyze the genetic similarity of the virus with dendrograms, homologies, and key sites to understand what its relatedness is to the human strains as well as our other swine-specific strains. We can take the same tools and technologies to then apply it to other diseases as well as other strains. So in conclusion, thank you all for your time. We went through all different technologies from first generation of most of our traditional vaccines that we're all very familiar with to second generation that primarily recombinant vaccines. Our third generation, the RP vaccines, as well as the RNA COVID-19 vaccines. And then lastly, we concluded with the Socovity dashboard, the tool set that we have to analyze and track pathogens and follow them as they're emerging and moving throughout not just the US, but also the entire world. Thank you. And with that, I will take any questions.
Channing, are you on?
Yes, I am.
Okay. Do we have any questions? Hi. There you are. Okay. Do we have any questions? Is there any confusion?
Hello, everyone. My name is Matthew Scott. I'm a DVM and...
Sorry.
Nice to meet you.
Sorry about that.
So,
What question?
So we are getting a few questions in over here. Would all RNA vaccines be 2 doses?
All of our RNA vaccine is a 2-dose vaccine. I think when you're using all of them that I'm familiar with, yes, are 2 doses, as well as the mRNA vaccines that are coming out with COVID. We have done some studies actually with FMD and classical swine fever, where you've been able to achieve a protective dose with 1 dose, but you need a incredibly high amount of RNA particles in order to be able to achieve that. And the immune system just is better primed when you give 2 doses. So I think it's potentially possible, but you definitely get more of an optimized immune response when you give it in a 2-dose. I can't speak for the mRNA vaccine because I'm not familiar with all of their data, and human data isn't exactly shared as openly as we are on the animal health side. So I can't speak to that for sure. But in general, this class of vaccines, I would say yes, typically are 2 doses. And we get the advantages of generating a CMI response with it, but yet it's not quite the same as a live infection because you are generating the antibody responses through just the stimulation of that gene of interest, not with the actual virus that it is being the pathogen you're targeting.
With the RNA and the messenger RNA vaccines, is the host immune response directed at the RNA sequence, or is it expression of the RNA within the host to generate the proactive anti protein?
Yeah, so with the RNA particle vaccine and what I've read about the mRNA vaccines is that the sequence and the RP or the mRNA and the nanoparticle is actually taken up by the immune system cells. Ours are typically taken up by dendritic cells, and they are then broken down and presented on both MHC1 and MHC2. And then the immune system is then presented those bits of protein and that's how the immune, so they're basically expressed to the immune system, and that's how it's then generated. So it generates protein basically, which then generates the immune response. So it's not going to be directly to the actual component in the vaccine. It's going to be then you basically use the animal's immune system or the human's immune system to basically use their own immune system to provide the actual response. Hopefully that answers your question.
So I have a simpler question.
Yeah.
Will this work on like toxins, like for tetanus? And then the other question is, if you were looking at like vaccines for ticks or for worms, could you use part of that the genetic makeup of that organism to do the same type thing?
Yeah, so some of the other products that we currently kind of play with is some toxin genes. So for Clostridium, we have Clostridium difficile and perfringens. The challenge with bacteria in general is that you need to typically do to the toxin itself and not the actual bacterium. And so you're not actually preventing the infection, you're preventing the damage that's being caused by the toxin. And typically when we're targeting viruses, our goal is to prevent the infection if at all possible. Typically these types of platforms, you only have so much room of the gene to be able to insert it in. Bacteria are large compared to viruses. So viruses tend to play better in these systems than bacteria or different parasites and stuff. The other big challenge that you have when you're looking at different types of viruses or bacteria or really any pathogen is what is that gene of interest that you're going to put into this type of platform. You have to know what the gene is that's going to potentially be protective. It's easy when you have a simple virus like a coronavirus that has a spike protein. That's a pretty obvious target. Obvious to researchers that are smarter than myself, to be able to put it into a vaccine. And like I gave the example of our PED, we had originally thought the S1 portion of the spike gene was going to be enough because literature had shown us that was going to be enough. And this whole spike gene is really large and it's hard to get into our system. So we just put in this S1 portion of the spike gene, and it didn't work as well. So then we were able to get in the entire spike gene, and once we put that in, that was a lot more efficacious. So it's figuring out what that gene is. Can it fit into the system? Do we get good expression? And it's really just knowing, it's also when the animal presents that immune system to it and makes the antibodies and generates the CMI to it, is that the actual protective response that's needed to provide protection against that disease. African swine fever would be a example where we don't have it figured out. That's a really, really complicated virus that has, hundreds of genes. And we don't know which one is the target that is remotely going to provide protection. So we don't know where to start with that one, honestly. We've tried, I think, 30 to 40 different genes and in different combinations, and we don't have the magic bullet yet. So it's just kind of honestly a guessing game. But the cool thing about it is you can manufacture, 40 different vaccines kind of at a drop of a hat, and try different things, and super safe as well. You're not actually working with the pathogen ever. You're just working with the sequence. So to me, it's kind of a cool way to work on, really risky pathogens, in low biosafety containment too, as well.
All right, so we do have another question. What about the DOI on these vaccines? Duration of immunity.
It's very variable by the actual antigen you're looking to protect. And the way we're regulated is we're under a prescription vaccine. And so there is because we're targeting pathogens like influenza that are constantly mutating and changing, we don't ever really go typically for fully licensed products that have efficacy claims with DOIs. So we don't usually do DOIs on those types of products. we would have, I mean, we have DOI data on some of our flu stuff that would be up to 12 to 14 weeks. So we know that immunity is there. We will actually get to the point where you can't measure antibodies anymore, but when we challenge those animals, they're still protective. So there is a CMI component and a memory antibody component that's there. The challenge is what we don't know so much about the immune response is, we all understand antibodies because it's what we can measure. It's what we can send serum to the lab and get results for. But there's an entire branch of the immune response that we just can't actively measure in the field, and that's the cell-mediated immunity. And I think that's a really, really important arm of the immunity that we don't understand. And our killed vaccines don't do usually a typically great job at stimulating that portion of it compared to live virus infections or our modified lives or these third generation vaccines. So I think we need better ways in the field to learn how to measure that. And we've started doing some interferon gamma measuring to be able to start to learn what that measures, look like, what they look like post-challenge, things like that. So I can't tell you what the DOI is. We've had animals that are coronavirus vaccinated for PED that, have seen the virus, and we continue to boost them for years later, and they're more protected than animals that had just seen the virus that haven't been vaccinated. So, but I can't tell you it's a year or it's 3 years. We don't also keep pigs around very long in our industry. You guys keep them around a lot longer in cattle. So, we don't really have that much of a problem with DOI on our side as much as you guys would. So it'd be a lot different and something we'd have to look at.